Assalamualaikum and good day everyone. Today I'm presenting my paper titled Intra and Postoperative Cell Salvage of Shed Mediastinal Blood Reduced Blood Postoperative Blood Transfusion with no additional risk of infection. This study was conducted in University Medical Center with the cooperation with Cardiothoracic Surgery, Infectious Control Unit and perfusionists. Cardiac surgery is the largest consumer of blood product. Allogenic blood transfusion carries substantial adverse effects such as allergic reaction, infection, TRALI, and volume overload. In a paper published in Annals of Thoracic Surgery, 48% of cardiac surgery patients receive PEG cell transfusion during perioperative period. Intraoperative cell salvage with a mechanical device, which also called as cell saver, is a well-established blood conservation strategy to reduce allogenic blood transfusion during cardiac surgery. The usage of cell saver also expanded beyond the intraoperative period, where post-operative chest drain were connected to it. The shed mediastinal blood was then washed and transfused back to the patient as an autologous transfusion. So the question is, does it really reduce the blood transfusion post-operatively, and does it increase the risk of infection? A paper published in ICBTS in 2011 shows that intraoperative use of cell saver alone does not reduce the rate of allogenic blood transfusion. This is because in the paper, they noticed that in less than 7% of cases, the blood volume loss was sufficient enough to be washed and returned. A paper written by Permagine et al. Reported no significant difference in allogenic blood transfusion even if it is used post-operatively. However, this paper only examined 99 patients, and it's also involved valve surgery where a lot of saline is used during the surgery, which can contribute into the hemodilution of blood during CPB. So the objective of this study is to investigate the reduction of allogenic blood transfusion in a patient who received a cell saver and it was used post-operatively up to 24 hours. And we also investigate the rate of infection in the cell saver group. It is a retrospective study. Patient data who underwent elective surgery from October 2019 to September 2020 were examined with the period of 1 year. We start to use a lot of cell saver as part of our blood conservation strategy due to blood shortage caused by COVID-19 pandemic. This is a picture where the usual setup of patient who had a cell saver in our CICU. As you can see here, the chest tube was connected to the reservoir for blood collection, and the reservoir is connected to a vacuum which is incorporated by the machine itself. And the collected blood will be transferred to the centrifugal bowl where saline is added and the blood is washed. The water will be sent to the waste bag, and the PEG cell will be transferred to the blood bag and ready to transfuse to the patient. We use SPSS question 23 as our statistical tool. The data were analyzed using a chi-square test. A p-value of less than 0.05 was considered statistically significant. We had 158 patients who had an elective CABG, and 41% received cell saver, while 58% does not. So this is the data and the variables that we examine in the study. There is no statistical significance in all of them. There is no significant difference between the two groups in a patient: age, gender, pre-op HB, CPB, and cross-clamp time. And we also examine the fluid added during CPB, hemofiltrate volume during CPB if it is used, and the urine output during surgery. So the result is the blood transfusion reduced dramatically in a patient who received a cell saver, which is 36.4 versus 66.3 for a patient who does not receive a cell saver. Average blood transfusion received is much less in the cell saver group compared to the patient who does not have a cell saver. Other result that we examine is the rate of blood transfused, 4% in cell saver and 10% in patient who does not have cell saver. Chest reopen 3 versus 5%. However, both of them does not have a significant difference. For the suspected infection patient, there is no significant difference. It is 21 versus 23%. Suspected infection means that if the patient have an increase in white blood cell, raise in temperature, require escalating of the blood of the antibiotics, and there is no significant difference between two of them. And there is no significant difference in positive blood culture between the two groups, and also mortality. So the conclusion is post-operative cell salvage of mediastinal shed blood using cell saver reduce the rate of allogenic blood transfusion with no additional risk of infection. With that, I end my presentation, and thank you for all my colleagues who involved in the study from University of Malaya Medical Center. Thank you.